Okay, great. Okay. I think we're set. Cool. Yes. Thanks a lot for inviting, for introducing me. So my name is Dmitry. I work in Germany, in Tübingen, and I'm interested in neighbor embeddings, in contrastive learning, in various ways to visualize complex data. That's what I'm going to talk about today. So let me just very briefly flash these images to so that we're on the same page about what I mean when I say visualizing complex data. So the setup we're working on is that we have some objects, some collection of objects. This may be coming from biology, like single cells. Everything that you see on top, every dot is a cell, but it could be images, like in some of the pictures in the bottom, or it could be texts, documents, something. You have a large collection of objects that you want to study. You want to explore it. And for that, we argue it's very useful to make visualization like that allow you to either spot something interesting, maybe something unusual about the data, maybe something you didn't expect that you could later go on and confirm, or maybe there's something fishy in the data, something that looks like an artifact. So you could do quality control of your data, very valuable in biological context, for example. And in fact, there is some debate, or there could be some debate, maybe we'll talk about that later, whether visualizations, like two-dimensional visualizations of very complex, very high-dimensional data, is a useful tool at all, because of course there's a lot of distortions when we project 1,000-dimensional space to 2 dimensions. This is inevitable. My point of view, and something that I will try to convey in this talk, is that this is nevertheless useful. Despite all the distortions, this captures, this kind of visualization can capture important aspects of the actual high-dimensional data that we can't observe directly. Okay, so the talk is going to be roughly divided in 2 parts. In the first part, I want to talk about how these things work. The neighbor embeddings, what I mean when I say that it's related to contrastive learning, and all the mechanics of that. And in the second part, I will focus on this particular thing that you see down here, which is our visualization of PubMed, which is something that we shared very, very recently, just a few days ago, and I'm excited. So I will present that as a use case. Okay, so part 1 is contrastive learning and neighbor embeddings. And I'm going to start with this diagram. So I've never shown it to anybody. I made it for this talk. So let's see if it's actually understandable or not. This summarizes my entire thinking about visualization at this point. So we have here a collection of objects, and this is very general. So these could be points in high-dimensional space. So we have 1,000-dimensional space, and then there are points. We want to know if there are clusters or maybe some continuous structures there. So we would like to represent these points in high-dimensional space in a 2-dimensional space, for example, just on a sheet of paper, right? So every point gets mapped to a point here. So when you're using tools like t-SNE or Umap, things that are called neighbor embeddings, this is what is happening there, right? And all of those methods find neighbors in a high-dimensional space and try to embed them in 2D so that the neighbors stay neighbors. So there is this underlying idea that the similar objects, in this case neighbors, should stay close to each other. But now comes the interesting part. Let's say your data is a collection of images or a collection of texts, or you could have a graph and your points are actually nodes of the graph. And I still want to represent them in 2D so that the similar objects are close to each other. But what does similar mean? So the word similar does a lot of heavy lifting here. Similar here just means Euclidean distance is small and it's a neighbor. But what is similar for images? I can't take distances in pixel space. So there is this very powerful idea of contrastive learning, and I'm going to explain it later in more detail, is that we can actually generate similar images, pairs of similar images, and we will use that to train the model. And we could do the same thing with texts, actually. And for the graph, similar nodes means that they're connected by a graph edge. So there should be some way for you to define similarity on the objects, or maybe generate similar objects. So if you can get paired objects, that's the only thing we need here, then we can embed them so that the similar objects stay close to each other. And there is another important distinction. whether we do this in a parametric way or not. So parametric means there's a function. There's a function from 1,000 dimensions to 2 dimensions. It could be a neural network that we could train, or it's a function from the pixel space and it's a convolutional network to some output space. In t-SNE or in the Umap, the methods that I think most of you have heard about, there is no function. It's not parametric. So we could have these 2 settings, and the output space could also be different. So in t-SNE and Umap, it's usually just R2. In computer vision, in the modern methods for contrastive learning, unsupervised learning, self-supervised learning, as it's called often, in computer vision, this is usually a high-dimensional space. So it could be constrained to a sphere in 128 dimensions. So the purpose here is not to visualize, actually. It's to learn some representation of the data, but the focus is not on visualization. My personal interest is on visualization, so I much prefer to work with this output than this 128 dimensional output. And I'll show you an example later of how to go from here to here. So this is very generic setup, and many different things, from t-SNE Umap to SimCLR to graph contrastive learning to sentence BERT, I think somehow fits into here. And we'll have some examples throughout the talk. So let's start with just t-SNE Umap, neighbor embeddings. And this example uses MNIST, so a data set that I think most of you are familiar with. So that's handwritten digits. Each object is this little image, right? There are 10 classes. 10 digits. We want to visualize this in 2D. I can do PCA and color by the digits, and it looks like a mass. Everything overlaps. One can do something more sophisticated, like multidimensional scaling, a technique from 50 or 60 years ago that tries to position points in 2D to preserve the distances between them in high dimensions. It also fails miserably. Everything gets mashed together in this one blob. And then a t-SNE does this very nice visualization, arguably very nice, that actually separates all digits. The classification accuracy in this 2-dimensional space is like 98%, and one can see even sub-digit structure in some cases here. So one interesting question that I think we should ask ourselves when we look at that is, why does MDS fail? So that's interesting, actually. So that's the loss function of MDS, is we have some original pairwise distances in 700 dimensional space, the input space of MNIST. We want to position them in 2D. We want to match Euclidean distances. It sounds very sensible. Why does it not work? The reason it doesn't work, I think, is that if you look at the distribution of pairwise distances in high dimensional MNIST, they are all large. This is one manifestation of the curse of dimensionality. There are no small distances in that space. All distances are large, and in some sense, they are all similar to each other. So they are all, like, the scale doesn't matter. What matters is that this distribution is far from zero, right? It's like shifted away from zero. So you can't possibly arrange points in 2D to have this distribution. So MDS cannot succeed, so it has to somehow break down, and in the end, we don't get a useful result. The really groundbreaking idea, I think, of neighbor embeddings was to say, let's not do that. We're giving up on the idea of preserving distances, but what we want to preserve is very small distances. We will find neighbors in high D, and these are things with small pairwise distance compared to the whole distribution, right? And then we want to preserve those, only those. So we disregard most of this distribution, just forget about it, find nearest neighbors and want to position them close. This was really, so this was, as far as I know, first suggested in 2002. That was not t-SNE yet, that was SNE, but that's the core idea, and it didn't change since then. Everything, Umap, everything that's popular now uses this. So that's the stochastic neighbor embedding idea. You construct k-nearest neighbor graph, so you find neighbors, a very small number of neighbors, like 10. Then you have pairs of neighbors. You say that everything that is a neighbor of each other has like an affinity, it's called, which is not zero, and everything that is far away from each other just has zero affinity. And then you want to position points in 2D to minimize this loss. The details don't really matter. Maybe you've seen that the t-SNE loss, you want to minimize the loss where the affinities in the 2D are defined such that the points that are close, that's the distance between an embedding, they have high affinity. So the neighbors get attracted, and not neighbors get repelled from each other. They get repelled because everything is normalized to sum to 1. There's this normalization constant over here, so that we can think about P and Q context. See, you just divide by all pairwise distances, and this, if you take the gradient, then it's easy to see that this results in this repulsion overall. Everything gets repulsed. So this can be illustrated very easily if I just show you the animation. I find this very, very intuitive. So this is gradient descent of the t-SNE loss function on MNIST. Every point is a digit. I will let it run a few times. It starts from a random configuration, and what you see is that neighbors attract each other, and this forms these clusters because neighbors of the same, like neighbors of digit 5 are very similar written digit 5, right? So digits 5 start to attract each other, and you get these blobs forming and moving around. What is interesting, in particular for my talk, is that this is actually far from optimal. So it converges to something, but it is stuck in a local minimum, in a bad local minimum in some sense. Like these red points over here want to merge with this or connect with these red points over here, but they cannot because there is something else stuck in between, right? And it shouldn't be there. Ideally, we would like to move these blue points over here. This would decrease the loss overall, but it's stuck. So a very powerful trick that was suggested already in the original t-SNE paper was to do something that they call early exaggeration, just increase all attractive forces for some time and then switch it off. And this makes essentially these separated red blobs sort of penetrate the obstacle, you know, tunnel through the wall and merge together. And then you switch it off and then you get the final result. So let's look at it once more. You will see here that in the beginning, very fast, these clusters form. And then at some point, these exaggerations turn off. You saw it now, everything sort of, there's additional repulsion, right? And everything, like you get to see more inner structure within each digit. So that's standard t-SNE. That was sort of introduction. Now I come to interesting parts of this. So one thing that we were really wondering about for a while is when you look at this optimization and you look at what happens during this phase with increased attraction, the result that you get actually looks often surprisingly good or interesting in its own way. So here we have t-SNE with like standard t-SNE of MNIST, and this is the result that you get with somewhat increased attraction, stronger attraction. So we can get the whole spectrum of embeddings where we vary the force with which, the strength with which the neighbors attract each other. So if they attract each other stronger, you get larger structures forming. And notice here, for example, this structure over here includes 5, 8, and 3, and 5, 8, and 3. Well, they share a lot of pixels actually, right? There is a circle in the bottom, or half circle. So it makes sense that they feel some attraction. And if you increase attraction overall, then you see these larger groups forming, and you see a lot of white space in between. So that was interesting for us. And then we quantified across a whole bunch of data sets that this spectrum is meaningful. So this is the work of my student Nick. This spectrum is meaningful because if your data have some continuous structure in it, so this is just to illustrate a simulated data set that looks like a necklace. So we have Gaussian blobs that are arranged on a line and sort of touching in high dimensional space, I think in 100 dimensions. So if we do t-SNE, you see the blobs very nicely, but you don't see the arrangement, this line continuous arrangement. If you increase the attraction strong enough, you see that this is in fact a one-dimensional manifold. This is very relevant in biology, where people often look at developmental at the development during just biological development in the fetus, for example, and then one can see how cells progress in their cell state. And that's often of biological interest, and this will be obscured if you just do t-SNE normally. And another very interesting aspect here is that, so some of you may have heard of Umap, that's like a method from 5 years ago or so that's often seen as a competitor to t-SNE. we realized that Umap gives a result that is very, very similar to what we can get with t-SNE if we increase exaggeration to something like around 4. But we tried a lot of data sets, Nick tried a lot of data sets, and it always works. So it's not a coincidence. It's Umap is really working like t-SNE with increased attractive forces. But why is that? So at the time when we wrote that paper, we thought Umap uses a technique called negative sampling. I don't have time to go in a lot of detail about what it means, but in practice, it means that it just samples 5 or small number of repulsive forces per each attractive force. So there's a pair of neighbors. You take one during each gradient descent step. So you have one point and another point that feels an attractive force. And instead of repulsing to everything else, you just repulse to 5 randomly chosen points. That's what Umap does. And we thought, okay, of course, this reduces the repulsion because instead of repulsing to 70,000 points, you just repulse to 5 points. We later realized that actually t-SNE can also be optimized using this sampling technique where you only sample 5 repulsive forces on each iteration per each attractive force. And this was recently suggested. So one can do that by using something called noise contrastive estimation. And this left us very puzzled after this paper was published already about how then, what's the difference? And this was, I think, very nicely explained by Sebastian in a follow-up paper that was recently accepted to SimCLR, where Sebastian studied this this family of optimization methods called noise contrastive estimation, NCE or info NCE. These are different flavors of this noise contrastive loss. And just very, very briefly, the gist of it is that instead of computing this very computationally expensive and squared interaction between all points that generates the repulsion, right? We just sample few random repulsive forces, and the magic of NCE and info NCE is that it still works and converges to the same thing. One can prove that. That's the theory of noise contrastive estimation. So one can run t-SNE like that. That's another way to do an effective t-SNE implementation. Umap does something slightly different. It uses negative sampling, which is basically a version of that, but does not take care of learning this Z constant. And Sebastian could show that this introduces the bias towards attraction. So we could regulate this attraction-repulsion spectrum where you are on the spectrum by regulating the parameters of this estimate. Why I think this is important for the bigger picture of my talk today is that this allows to do this approach to this noise contrastive estimation approach allows to do things that we can't do with standard t-SNE. For example, what if we want to do parametric t-SNE? What would parametric t-SNE be? Let me remind you of this image. So we start here and we want to get here, but now I want to have a function, which I will choose as a fully connected neural network. To train a neural network, I need to select mini batches. So everybody who trained a neural network knows that it uses stochastic gradient descent. But if I only sample some fraction of my neighbors, then I can't compute this Z term that actually is a sum over everything. So that's a problem. So there actually has not been any implementation of parametric t-SNE for that reason, I think. But using this noise contrastive estimation, it's super easy. It's super easy. Sebastian has this PieTouch framework, which is actually in some sense simpler than the original t-SNE implementation because PieTouch just does everything for you, right? Automatic differentiation and so on. And we can just use either non-parametric, for example, t-SNE with info NCE loss, or parametric t-SNE with info NCE loss. So in this case, we have a neural network that performs the mapping, and it's the same loss function. It's basically the same code. Just need to replace the model that is being used. So it's very, very convenient. And moreover, if you do t-SNE with the parametric function, with the info NCE loss, then you're very close to something from seemingly entirely field in machine learning, in computer vision, something called SimCLR, and this is the contrastive learning part. So the way SimCLR works is that we have for each input, this is now image datasets, right? I will show an example in a second. So you have an image. You don't want to find its neighbors in the pixel space. That's a bad idea. So we can't use t-SNE on images. The trick, that's really the main trick. We take each image, we perturb it twice by just shifting it, for example, rotating it, changing the colors. And then we can say, well, these images that were generated from this one, so they are similar. We know that this is a pair of similar images, so we can push them through the network and make the contrastive loss, pull them closer. Okay, so that's SimCLR. It's very successful in computer vision, but never really used for visualization, just used to map everything to 100 dimensions for some kind of downstream task later on, maybe classification. But we want a visualization, so what do we do with that? So here's another more recent Nick's work. This is very simple this is a simple data set as long as image datasets are simple. So if you throw that into t-SNE, then you don't see any structure at all. There are 10 classes. So in some sense, it's similar to MNIST. 10 classes, the size is the same as an MNIST, but t-SNE can't do anything because neighbors in pixel space are meaningless. So we want to do, or Nick developed the algorithm that we call t-SimSNE, which is basically SimCLR, but with two-dimensional output. And the trick is that if you just do naively SimCLR with two-dimensional output, it works very poorly. So we needed to, or Nick succeeded in developing an effective training strategy that does that. But the core idea, so I don't have time to explain all the tricks, but the core idea is the same. So each image gets perturbed twice, goes through the network, ends up somewhere in 2D, and then we quantify how similar points are to each other using t-SNE's Cauchy kernel. So it's sort of t-SNE meets SimCLR. It's a merge between the two. That's the influence loss function. We can train it, and in the end, we get for cipher 10 this embedding. And I actually just want to show you how meaningful it is. I'll try to convince you. So this is around 70,000 images. They're split in 10 classes. These are colors. But if we look at the image that we got, we were actually, at least I was surprised. I didn't expect this result, to be honest, because a lot of the classes, so birds, green is birds, and a lot of them, so birds, this class got split into several parts. And in fact, this happens for almost each of them. So we were puzzled at first, but then since it's images, it's very easy to look at them. And we see over and over again, basically in every single time, that it's very meaningful. So this blob is bird heads, like close shots of bird heads. And these are just birds, small birds sitting on a tree, you know, photo like that. And these are huge birds like camels. So these images do look different. They arguably correspond to different qualitatively different things, and this is picked up just with our method completely automatically. So the horses split into the horse and mounted horses, or horse with a rider, and so on and so forth. There's one little aspect that you won't notice otherwise. This little thing. So if you looked at a lot of t-SNE or Umap plots, then this looks suspicious. Like, why is this so dense and somehow looks like a stripe? This should be a red flag for you if you have experience looking at these images. So of course we looked what it is, and we realized that it's just the same image over and over again. It's not absolutely identical, but it's different photos of the same car from the same angle. I'm not sure why this is part of this data set, but there's over 100 images that look like that, and this just gets super prominent in this visualization. In this particular case, this is not very useful, but arguably this is an example of what I say when I say that one can use it to spot inconsistencies in your data. So maybe you want to, if you have some application, then you may want to exclude such images. Okay, I have example with cipher 100 dataset, which is dataset of the similar size, of the same size, but has 100 classes instead of 10. And well, just believe me that one can see a lot of structure here. And I'm like here, baby, woman, girl, and man. All these classes actually overlap, so our method is not able to separate these classes. But the images are also really, really tiny. It's actually even hard to separate a boy from a girl sometimes, even for a human observer. even though I think we could do better in principle. There are interesting things like flatfish is sitting in the same island as the people. So why is that? But if you look at this, it's actually always a fisherman standing and holding like a huge fish. So these are the images. They are labeled flatfish here. That's their class, but in fact, they could as well be labeled a person. So this picks up structure, even in the cases where you think the colors may suggest that it's not ideal. Actually, sometimes it's the labels that are not ideal, but semantic representation is correct. So we were very happy to get that. Okay, so I almost took most of my time, but I still want to, maybe I have 10 minutes to talk about PubMed part. So this is switching gears a bit because PubMed is not using this contrastive learning. This is just back to normal t-SNE. I will in the end say how I think these two things may come together. But this specific preprint that we just released, so that's led by PhD student Rita, and also in collaboration with Ben Schmidt, who is working in a startup in New York in Nomex. I will show you later what he did. It's really cool. So we wanted to visualize PubMed. So maybe some of you know this database and some don't. So it's everybody in biology uses this all the time. It's National Library of Medicine in the States, collects metadata on all scientific publications and everything that's remotely related to biomedicine. It's very inclusive. So they say here that they have 35 million papers in there, but in fact, some are don't have full metadata. We wanted to work with abstracts. So the abstract text, you don't have full text in PubMed, but you have the title, the authors, and the abstract mostly. So we only selected English language abstracts, and this left us with 21 million. But we took everything they had, PubMed had. So there's 21 million English abstracts in PubMed on all kinds of medicine and everything in life science, biology, neuroscience, and so on. And we thought, and this originated as basically a fun student project, can we use the neighbor embedding methods like t-SNE to visualize this entire corpus of literature? I think this is the largest t-SNE in a sense, like it's embedding with the most points that at least I ever worked with. It's uncommonly large, let's say. So what we did here, and maybe if there are questions, then we can discuss later in more detail. But we take each abstract, we pass it through a language model that we didn't train. We just selected a language model that worked best for our purposes. In this case, it was a PubMed BERT. So what this does, it takes a text and outputs a vector in 700-dimensional space. And then we take those 700-dimensional vectors and put that into t-SNE to reduce it to 2D. And that's our final embedding. And what you can see here on the left, it's colored by the journals it's published in. I know genetics in it, like the Journal of Genetics. Then that gets assigned to the class genetics. So we assign the labels here in a very crude way, just to have some way of actually coloring this embedding, or also seeing how well we can separate these fields. So this is based on journal names. This is the same embedding colored based on the year of publication, and some things are really prominent here. For example, so the yellow is modern papers, and if you look at it, then you quickly notice that there is this yellow blob, right? So it's one cluster of papers, and this cluster is very, very recent. Everything is published in the last 2 years, so of course it's COVID, right? It makes sense. Yes, so we can get back to this if there's interest. I will just say that Rita tried a lot of different language models, and we selected the one that works the best. And by works the best, this separation between fields is the best. So what we were interested in, or how we quantified the quality of our embedding, is how well cardiology separates from radiology. You know, that's a quantitative, that's a number. So with all kind of different language models, with all different t-SNE parameters, we could see how well we're doing. And that's the pipeline that was optimal in our hands. Like we couldn't, that's the best results we achieved. That's the language model that we used. And then later on, we used t-SNE. Also, one needs some care to be able to run it actually on 20 million. If you just use default parameters, we run out of memory, despite having a really large computer. So there are tricks of how one can do it more memory efficient, and then it works, and then you get this result. So in the last 5 minutes, I'm going to walk you through several examples that we have in the paper, and I'm happy to talk more about that afterwards, of trying to convince the reader, you, the audience, that this embedding can provide some meaningful insights into the library, into this collection of papers. So we have several examples. They are sort of disconnected from each other because it's more like case studies. So this is, by the way, zooming in the COVID region here, right? And coloring now only COVID papers by papers on the vaccine development, and there are COVID papers on the, I don't know, mental anxiety that people feel because they don't want to go and take a vaccine. And one can see a lot of structure here that nicely separates. So if you actually look at what papers are in one of these little islands, then they are all similar. So it makes sense. There's structures preserved here on a, not only on this big course level, but also on a pretty fine level, actually. And we could also show that COVID forms uniquely dense cluster compared to some other things that we would could look at, like influenza. If you select all influenza papers ever published, they are not as tight a cluster compared to COVID. So that's interesting, that the pandemic basically transformed, or at least affected, the entire publishing, biomedical publishing, so that this cluster emerged. Another example here, maybe I almost skip it in the interest of time, but there was something interesting for us because we work a lot with neuroscience, that if you look at neuroscience, then this is the neuroscience cluster, but also this in the bottom is the neuroscience cluster. So we were like, it's interesting, why is it split in two parts? So we looked into that and realized that on top you have things like cellular, molecular neuroscience, and in the bottom you have more cognitive and maybe computational neuroscience. So both are called neuroscience. Both kinds of papers can be published in the Journal of Neuroscience or Nature Neuroscience. So they're labeled neuroscience in our hands, but actually very different papers on very different topics. And interestingly, we could also see that older papers are on the top, or there's more old papers on the top, in the top cluster, compared to the bottom cluster, and more, of course, there are recent papers here and old papers here, but on average, you know, this is more modern research. So we could actually directly quantify that overlap in neighbors between neuroscience and, for example, psychology grew in time, and overlap with things like pharmacology and something else that is also on top of the embedding decreased over time. And this is, by the way, something important for us, because whenever we see something in the embedding that looks like, oh, that's interesting, ideally we want to go back to high dimensional data or to the text or to something and confirm that. Because very often people say, and rightly so, that the embedding can be misleading. Maybe it's split in two parts, but it just failed to converge. Maybe it's also a bad local minimum. You know, one can worry about that, and one should worry about that. So the approach that I don't know, preach about is that once you see something interesting, try to confirm, convince yourself that it's real. But the point is that looking at the data like that can give you inspiration to actually start asking these questions. Okay, so more case studies. We looked at where different machine learning methods were used. And again, we only working with abstracts here. So this is every abstract that says convolutional neural network in the abstract gets this color. And we can see papers using linear regression and factor analysis, or random forests, or deep learning, or support vector machines. And then we also looked at which papers mention machine learning in the abstract. Like they write, we use machine learning to do something. And well, that clusters very strongly. So there are fields in biomedical research that self-claim to use machine learning. So we sort of annotated them manually, and then one can say, one can quantify things like, okay, in the research on protein structures, which methods are often used, and then we see support vector machines. You can't see this here, but we have a table that directly quantifies that in the paper. For example, support vector machines are strong here, and neural networks as well. So it makes a lot of sense, but it's interesting, I think, to see this parts of the research community that use machine learning. Okay, I have two more examples, and then I'm done. So this is the gender bias in the biomedical publishing. So of course, this is not a perfect quantification of this, but what we could do with the data we have is that for each paper we can get the first author and then take the first name of the first author and then look up in a big database of male names and female names whether this is likely male or likely female name. And we could do the same with the last author, right? For some papers, especially older papers, we don't have the first names, we just have initial and the last name. There we can't infer the gender. But for most papers we actually can. And this is colored by the gender of the first author and last author. So one can see that these parts are like STEM fields, physics, and more math. So this is more male dominated. This looks blue, right? And this is more psychology research, for example. So that's more orange. But there's actually all kinds of things, and different medical specialties have different gender ratios. We can see that it grows in almost every discipline. The fraction of females grows with time over the last 20 years. But what was most interesting for me is that you could in some cases zoom in, look at things like surgery. Surgery for some reason is very male dominated, at least the authorship in the surgery papers. So this island, I think, is heart surgery or something like that. So it's very, very male. The fraction of males is very high. And then there is this orange island. So why is suddenly there is an orange island in the male field? Turns out that these are papers on veterinary surgery. And this happens in different, so we have many examples of that. Education splits by nursing training and medical training, for example, in the and shows different gender bias. So again, this shows that the embedding is meaningful on the scale of these pretty small clusters, and again also shows that it can suggest sort of meaningful questions for you. So if you just have a priori label surgery, then of course you don't need t-SNE to compute how many surgery papers have female first authors. But this would rely on this a priori choice of labels. Here we can just look at it. We don't need to have any labels. In fact, we just look at it and we see, oh, there is a blue island and there's orange islands. That's strange. The fact that they're close by means that the abstracts are kind of similar, share a lot of words, but the gender ratio is very different. Why is that? Well, and we see why, right? So it's the questions suggest themselves by looking at the embedding. Okay, and one can do really a lot of things, I think, in terms of exploring this embedding. So I don't know, also fun things like you could look at where people say little is known in the abstract or interestingly, so on and so forth. One last thing that I want to explain, and this is not in the preprint because we just found this a few days ago, actually, and didn't have time to update the preprint yet, but I find it super exciting. PubMed has information on retracted papers. There is something like 15,000 retracted papers in the PubMed. We can color, so they are still there. They have a normal abstract, everything, but they have been subsequently retracted. So we can color we can highlight them here in black, and it's very uniform distribution. It's a very non-uniform distribution. In fact, there is this island here that looks like everything is retracted. Of course, it's not. Most papers are not retracted. You can see on zoom in, but a lot are. So that's weird. At least that was weird to me the first time I saw that. Why is there a cluster of papers? That means they are semantically similar, and this has unusually high retraction ratio. And you may think, oh, maybe this is some kind of research in, you know, in one of these fields that was like a psychology, had problems with replication studies. It's something completely different. It turns out that most of these papers were retracted because people realized paper mills, organizations that would just write fake papers for you if you pay them. Apparently, most of these papers have Chinese authors, and that's a problem in Chinese research, in particular in Chinese hospitals. I was not really aware of that, but of course, there are people who are aware of that, and for them, this is maybe not surprising. What is interesting for me still, though, is there is this island. Most papers are not retracted. There is a very large proportion of retracted papers because somebody complained to the journal that, you know, there's two papers and they have the same figure, so this must be retracted, and sometimes it was. But I suspect everything here is not legit. I can't prove that, or at least I'm very sure that a lot of papers that are gray should be retracted, in fact, and we can just see on our map where these dangerous areas are that would require additional investigation. So I find this fascinating. Okay, I can finish up. So we have a website that was the collaboration with Nomex. So we have a website where you can explore that. It's fully interactive. You can click on every paper. You can zoom in like in Google Maps, and you know, papers load when or points load when you're zooming in. It's amazing. So Ben did that. And is still working on improving this, so one can explore this cluster of fake papers over here, or you can search for whatever topic is of interest for you and see how it looks. And just to finish, so this didn't use any contrastive learning, but in fact now, after doing that, I'm very interested in doing something that would take the texts here, put them through the transformer model here, and map it not to the 700 dimensional space, but maybe directly to 2D in the spirit of t-SNE. So we haven't even started doing that, but that's very interesting. Or also another question, can we take an abstract and somehow perturb it twice, replace some words or something, and then use the contrastive learning idea to say that these points should be close in the embedding? This is not used currently, right, at all. This is not working like that, but it could. So a lot could be done, I think, in all these modalities by bringing together these contrastive learning ideas and visualization goals. Okay, with this I can finally finish. Thanks to my collaborators and funders, and I'm very happy to take your questions.
Thanks a lot.